Let's welcome to part 4 of structure of DNA. Here we are going to study about different types of DNA and their structure. So as you can see in the chart, I have drawn this chart because it will take more time to draw during the video. So there are basic 7 characteristics of each kind of DNA. How many DNAs are there? Basically there are 5 kinds of DNA. BDNA, ZDNA, ADNA, cDNA and ddNA. Okay, so let's see different characteristics of this DNA. The discovered DNA by Mr. Watson and Crick, that is present in most of the living beings, is B form of DNA. Is which form? B form of DNA. Present in most of the cells. Present in most living cells. In most of the cells. Okay, so which form of DNA is predominant? B form of DNA is predominant, which is described by Mr. Watson and Crick in their 1953. So let's see the first characteristic. The first characteristic is handedness of helix, whether it is right handed or left handed. Likewise, the helix is twisted like a ladder. So on which side is it twisted? On right hand side or on left hand side, depending on that the handedness of helix is determined. So B form of DNA, which is discovered by Mr. Watson and Crick in 1953, is right handed one. Okay, what about ZDNA? It is left handed one, and all other DNAs, DNA A, DNA C and DNA D, are right handed. Only one Z type of DNA or Z form of DNA is left handed helix structure. Now let's see the second characteristics. Here, one turn of DNA pitch is calculated in length in the angstrom units. So one turn of pitch of DNA is 34 angstrom in length in B form of DNA, 46 angstrom in length, one turn of DNA, 46 angstrom in length in Z form of DNA, one turn of DNA 25 angstrom length in ADNA, one turn of DNA is 30 angstrom in length in cDNA, one turn of DNA is 24 angstrom in length in ddNA. So there is a considerable difference. Then if we want to calculate the diameter or want to study the diameter, there is still difference in diameters. So which one is having less diameter, which one is having high diameter, we will see. The diameter of normally present B form of DNA in each and every kind of cell is 20 angstrom. So how much is the width of this DNA molecule, B DNA molecule? 20 angstrom. Width of Z DNA molecule is 18 angstrom. Width of A DNA molecule is 26 angstrom. Width of DNA form C or C form of DNA is 19 angstrom. About D form of DNA, it is not mentioned very well in most of the books. That's why I have written it as a dash. Okay, now let's see the stability. Which DNA is most stable? The most stable DNA is obviously the one which is present in all the living cells or most of the living cells. It is B form of DNA. So most stable and physiologically active DNA is B form of DNA. What about Z form, A form, C form, D form of DNA? All these forms are unstable. These are unstable forms. Only BDNA is stable and physiologically active in most of the living cells. How many base pairs are there per turn? In B form of DNA, there are 10 base pairs per turn. In Z form of DNA, there are 12 base pairs per turn, but it is a dimer. It is a dimer or dinucleotide repeating unit is there, where in Z form of DNA. Then A form of DNA, there are 11 base pairs per turn. In C form of DNA, there are 9.33 base pairs per turn. In D form of DNA, there are 8 base pairs per turn. So as we can see, by diameter, the largest diameter having DNA is ADNA and the lowest diameter having DNA is ZDNA. And by number of base pairs per turn, ddNA is the smallest one, contains only 8 base pairs per turn, and the ZDNA contains 12 base pairs per turn. So it is by turn larger in length. Okay, so what is the turn length or base pair? The base pair is 12 base pair per turn, and in ddNA 8 base pairs per turn. Next, let's see the distance between two base pairs. So how much is the distance between two base pairs in B form of DNA? It is 3.4 angstrom. In Z DNA it is 3.8 angstrom. In A DNA it is 2.5 angstrom. In C form of DNA it is 3.3 angstrom, and in D form of DNA it is 3.03 angstrom units. Let's see the repeating unit. In all DNAs, the repeating unit is mononucleotide except the Z DNA. In Z DNA, the repeating unit is a dinucleotide. So you can pause this video and note down the details given in the chart on the board. Till then, bye bye. We will continue in the next video with some new topic.